Tu has hecho live.
¿Ah?
Tu has hecho live.
Nunca.
Yo tampoco. Eso va ahí.
Sí, y que escreve como Painting Outspace.
Can you see if it is recording? Good.
Deus.
Oh. Dá pra apoiar na garrafa.
Ah, tenemos laser.
I can get it with Stephanie.
Isso aqui? Olha esse vídeo aqui, velho. Yeah, okay. It's nicer. Like, I'm gonna look at that.
My ear is coming with the point.
I mean, it's good that I can pop the visor. Like, the visor and point. Here?
I guess.
Okay. I mean, it is what it is.
Puxou aí.
Like, oh.
Fica do lado aí, Bruno, pra tirar uma foto. Ah, é?
Agora?
Peraí, vou tirar outra. Outra. Manda pra mãe. É. Vai fazer live?
Uhum.
La one. Ya buscamos a la vez.
Tá pronto? Tem uma tarefa que tem que fazer primeiro nele. Só, resistir. Nossa, minha boca tá cansada. Elogio também, também Brasil. Também. Vocês mesmo que... Ai, eu tento fazer um fica bonitinho. Bora ver o dia, eu faço a foto. Eu aceito. Eu queria tanto te chamar. Eu não sei falar, você acha que você já desiste. Ai, eu não sei tirar cutícula porque a minha cutícula dentro do avião. I don't think.
Okay. Hello, everyone. Thanks for coming. This is our first graduate student seminar. So today we will have Bruno Scheffer, and he will be talking about plants in outer space, Star Trek. So a little bit about Bruno is that he's a first year master student in Dr. Quinn's lab. He came here before as a visiting scholar. And he did his undergrad in Brazil in the University Federal of Rio Grande do Sul. Oh, perfect. Yeah, that's it. So please enjoy his talk.
Thank you, Juliana. So good morning, everyone. Thank you for coming here today. I'm glad that Juliana already introduced me, but I just want to say that before somebody asking me, this unfortunately is not the top of my master. So it's just like a thing that I was always very curious about it. So I decided to do like a little research in the beginning. And that's why I came up with this presentation. And yeah, that's it. Okay, let's start with the reason why. Why exchange our beautiful blue planet for another one's inhospitable and lifeless ones? Well, there's a lot of reasons why. First of all, there is a possibility that another pandemic can come up on the planet Earth, and even sadly, an asteroid can wipe out life on Earth. It's kind of a crazy idea, but actually it is possible. So having like a backup plan is something really, really important. for any species that consider yourself smart. Besides that, there is a high possibility that in the process, we are going to find out answers to a lot of questions, such as, is it possible to colonize another planet? Is it possible to grow plants in another planet? Well, we probably will find out the answers for that in the process. And besides, testing new technologies that are capable of surviving these conditions are probably going to improve their technology that we already have here on planet Earth. Besides, we can mine metals that are scarce on Earth. And since the beginning of humanity, of humankind, we have this desire to push the boundaries of what we think is possible. So it's like innate passion about going forward to find out how far away we can go. So it's like a question like, why not? But for that to be possible, to travel, for example, from planet Earth to Mars, there is a huge space, a long space travel in the way. It, NASA says it's like 7 to 9 months to get there. So in the whole process to get there, the crew are going to find out that they have limited resource because they're going to need, like us, oxygen, food, mental comfort, a lot of things. And so we need to come up with a really, really good plan to match all of these needs. And it needs to be really almost perfect, because if you have, like, let's say in a system, 3% of waste, it's probably not going to be true that you're going to make it all the way, because it's 7 months or 9 months to get there, more this time to go back, and there is the time that you're going to spend in Mars. So we need to come up with a really good plan. So first of all, we need to map the human needs, and plants seems to be a key to this, sequestering carbon, filtering the air, producing food, and bringing psychological comfort to the crew. Nevertheless, there's a lot of challenges to this relationship to be possible. First of all, plants are evolutionarily adapted to the absence of cosmic radiation. But on outer space, that's not the case. And that can result to a lot of things, such as mutations. Besides, plants are adapted to the gravity stimuli to grow, to know in which direction to grow. In outer space, that changes. The gravity is different. So that can lead to another challenges for this growth of plants. So the question is, what is the impact of these two main challenges on the growth of plants? With that in mind, the objectives of this seminar are to discuss these challenges of growing plants in extraterrestrial conditions. So, like I said, radiation is not the same everywhere. On planet Earth, plants are adapted to receive just these kinds of radiations: infrared, visible light, and ultraviolet, because our mesopause layer and the ozone layer, it filters, it protects us and the plants against these kind of radiations. And when talking about ultraviolet, just UVA and B reach the plants, because UVC is completely absorbed by the ozone layer. But as we think about, okay, let's go to Mars, well, the plants are going to pass this layer, so they're not going to have the barrier anymore. So all of these kind of radiations are going to hit the plants. So what's the problem of this? Well, radiation, the main impact of radiation is on the DNA. And there's two ways that this can be possible. Through a direct impact, which would be the radiation directly hitting the DNA molecules. And this is the most hard one, because it's like a small target, but there's another possibility, more common, which is the indirect impact. On this situation, the radiation can hit another kind of compound, such as water, which is pretty common, and cause radiolysis, which is the dissociation of the elements inside this compound, delivering free radicals that are molecules with a missing electron. So these radicals can then hit the DNA and cause DNA damage. But we got a feedback system here, because the more radiation produce this ROS, which would be the reactive oxygen species, more DNA damage gonna occur, and more DNA damage, more stress to the plants because of a lot of reasons, and more ROS because of the stress. So it's like a bad cycle. And this cycle can up or down regulate a lot of gene expressions inside the plants. And this, don't figure out, is just a representation of this process of ROS production. And I'm just going to focus on the overview, not on every gene here. But first of all, this was an experiment by NASA testing the Mizuna plant on the ESS, and this is like mustard. And here they found out that the most important one is the OXY1, the gene OXY1, that kind of is the first one that knows about when there is stress. So the basically response of these genes is to make a downstream response to this cascade and tells the other genes inside this green line, which would be the MAPK cascade genes, like what's going on. So these genes tells the transcription factor genes to basically do something about it. Like, okay, we have this kind of stress, so we are going to produce this kind of protein to counterattack the situation. Okay, so this is actually the normal situation because we have the production of ROS and stress of plants here on the surface of Earth, right? In outer space, there is more up and down regulated genes. And this is represented by these red arrows here. So you can see that there's a lot of situations when this is down-regulated. And I'm going to give you some examples. First of all, OX1 can be up-regulated almost 38 times. And MKK4 can be up-regulated 5 times. And this gene can be up-regulated 15 times. So you can see how much more stress are occurring on the plants when they are in outer space, traveling, for example, from other planets. And again, this is a situation of a feedback system. More ROS, more DNA damage, and vice versa, which can alter gene expression. And I'll give you some examples here about the effects in the biological process categories in the plants. On the red columns are the up-regulated genes, and on the blue are the down-regulated genes. And on the x-axis is the number of transcripts, and the y-axis, the categories. So as you can see, there's a lot of important categories here that are up or down regulated. And I must say that this slide was like, this graph was like 20 categories filtering from almost 4,000 genes on the plants on this study. And just imagine, metabolic process, it's down-regulated all of these compared to the normal condition. So imagine a plant that is not capable of doing the normal metabolic process. So they're not going to produce enough for the crew. So it's a hard situation. But there's hope, actually. Even on the normal conditions of planet Earth, we got this mechanism which is called the DNA repair. So we got the situation of DNA damage, and this mechanism can bring it back to the normal condition. But even on this scenario, this mechanism can make mistakes and can do a misrepair. So instead of like DNA damage, like insertions, deletions, or single base substitutions on the DNA, these mechanisms can, instead of putting the right base can put another base or change the places again. So there is a chance. And this leads to mutations in the plants and genomic instability. So this is actually the normal situation. Again, on outer space, we got a lot of radiation. So radiation, it's competing against the DNA repair mechanism to hit the DNA. So there's a lot of DNA damage caused by radiation, but the DNA repair is not capable of competing against this mechanism. So there is more possibility of mutations and genomic instability, and even cell death is possible in this situation. So when talking about numbers, this is a representation of how much mutation frequency is possible in the x-axis in 10 million base pairs. And on the y-axis is the kind of radiations that are possible to be found on outer space, which could be fast neutrons, argon, carbon, gamma rays. And this kind of unit is called gray, which would be like how much of this radiation can be absorbed by a body. In a simple way, that's what it is. And the keV per micrometer would be how much of radiation it's leaved on the path that this radiation leads when it goes in a direction. Basic slides. Both are like a way to say the intensity of these kind of radiations. And between the parts, we got the type of material, which can be seeds or seedlings. And these acronyms are for the type of plants, like Arabidopsis, Arida sativa, rice, and tomatoes. Okay. And between the green lines, we got the controls, treatments. So ethyl methane sulfonate at 1% would be a substance that we already use to cause intentionally we want to cause mutations on the plant, so we apply this substance, but usually it's like 0.3% that we use. So in these situations, we want to find out like, okay, there's a lot of mutations on these plants that I applied this substance, and I want to filter which ones are the best. Mutations good for producing more food and other things. So this control is to know like how much far away the mutation frequency can go in a really bad situation. And this control would be in a normal condition, like spontaneous mutations that already occur on the surface of Earth and are really important to evolution, actually. Okay, so what you can see here, the main idea is that all of these kind of radiations have a greater mutation frequency when comparing to these spontaneous mutations on the surface of Earth. Besides, I must say here, in outer space, this is not predictable. All of these kind of radiations can occur at the same time at the plants, and the intensities can vary a lot. So it's a really hard situation. Besides, I want to ask you, from comparing this carbon radiation against the argon, do you think that just because there is more mutation frequency on the carbon is going to be worse than argon? Well, like a good agronomist, the answer for that is it depends. Because depends on the proportion of mutations caused on the plants. Because as I said before, there is single base substitutions in beige. There is deletions of less than 100 base pairs, which would be blue on this graph. And purple is the worst one, with more than 100 base pairs. And green would be insertions. So, as we can see, when talking about that example, we got much more number of deletions of more than 100 base on the argon case, as you can see here on this purple column. So it's actually worse for the argon, even though it has less mutations frequency. And the result of that is more loss of gene functions inside the plants. So this is represented by this red column. here, as you can see, there's much more loss of gene functions when talking about the number of genes mutated inside the plant. And there is more number of mutating genes with moderate impact too within the plant. So how are we going to do to get all the way there with limited resource, limited time to get there, everything, and still with radiation? It's hard, right? But it's not just that, actually. There's more. Gravity, actually, too, can impact the plants. On Earth, how it works? Well, usually the shoots grow in the direction of light, and the roots grow in the direction of dark. And the second stimuli is gravity. So the roots grow according to the vector of gravity, so in a positive answer. So this is how it works. This on the left is a schematic representations of the tip of the root. And these blue ones here are the columella cells. And within the columella cells, you can see that there is really small pink dots, which are the amyloplasts, and they're zoomed here. So this is a columella cell, and these are the amyloplasts. As the vector of gravity is this way, it's down, it's going down, these cells, they deposit at the bottom of the columella cells. So the plant knows, based on the same distribution of auxins on all the sides of the roots, in which direction to go. It needs to go down because the amyloplasts are down. But as we reorientate this tip of the plants, put it like this, like the example, the amyloplasts, they go down again. So the plants know that they need to go down. Okay? So this is a normal condition. Do you think it's going to be the same everywhere on planet Earth? Yeah, but gravity is not the same everywhere. So your Earth is 9.8 meters per second square, on Mars 3.7, on the Moon it's 1.6, and even microgravity is possible, which would be the gravity almost near to zero, which is possible like between two planets, when it's really far away from a spatial corpse, which would be like a planet or a star, things like that. So how it looks like? It's like this. So as we were talking about before, these red dots are the amyloplasts. So this is a normal condition with beat. And on normal gravity, they go at the bottom of the columella cell. But at microgravity, they don't know where to go. So they're like random distribution inside the columella cell. So how does it look like on the plants? On this trial conducted by this guy working with corn seedlings, on the left we can see a normal condition going up, right, towards the light. But when microgravity, the plants don't know where to go. They're like random growth to everywhere. And in a wrapped up seeds, white type seeds flown for over 10 days on the STS-9, which would be the space lab on outer space from the NASA. What we got here is the control on the middle, which would be with normal gravity on the surface of Earth. So we got a normal seedling with normal roots and normal shoots. On the left, we got thinner roots with very few secondary roots and twisted shoots. So it's the same gravity. Why do you guys think that, is it like that? So twisted. What is the another thing that can hit the plants on outer space that we just talked about? No? Radiation, thank you. So radiation is this treatment. Just radiation are inflicted on the plant. But on the right, we got both. Because in this situation, there was a machine capable of simulating the gravity on planet Earth. But on the right, It's gravity and radiation. So we can see a plant that don't know where to go. So again, it's hard, right? We got limited resource, we got radiation, and we got microgravity. Okay, what else, Bruno? Actually, there's another problem because there is the possibility of elevated CO2 on outer space. On planet Earth, this is how it looks like. We got usually moderate light supply. We got the ambient CO2 of 0.035% on the air. And the chlorophyll works well. There's photosynthetic electron transport chain and CO2 fixation. There's growth, everything's fine. But inside a spaceship, there's crew. They are producing CO2 because we are animals. And there's a possibility of elevated CO2, usually less than 0.5 because a question of health of this crew. But this is already 14 times more than the plants are adapted to. The plants are going to be
Someone is in the meeting.
well,
Maybe your mom.
I don't think that which is actually good on normal conditions on planet Earth because they're like storage, like food. But on outer space, there's imbalance between the highlight supply and how much carbon they are storing. So there's stress again. So it's another feedback system with production of ROS. And this inflicts on the food quality of the plants. As we can see on this trial, where on the green columns are the ambient CO2 with 0.035%, and the red columns are the elevated CO2 with 0.7, which is already 20 times how much the plants are adapted to. So what you can learn here is that when talking about protein, zeaxanthin, lutein, and provitamin A, there is a greater content on the normal environment than with when there is elevated CO2. And the result of this is less nutrients for the crew to survive this trip. So summarizing what we saw until now, we got limited resource and we got radiation, elevated CO2, microgravity, all inflicting on the plant cell. And this can lead to abnormal cells and even cell death. And the result looks like this. This is NASA's plants on the ESS in outer space. And we can see that it doesn't look so good, right? But nevertheless, there's hope. This is of a plant called duckweed, which can grow above water and can produce 20 times more protein than soybean. And this is the relative growth rate in microgravity in this axis, and in this axis the time in days. On the dark line is the microgravity, on the gray is the normal gravity. As you can see, this plant actually produces more in microgravity. Besides, it produces more starch content too in microgravity, almost double. So we got an unusual combination of multiply attractive traits in this case, because it tolerance against microgravity, elevated CO2, radiation, has a high productivity, it's easy to grow, it has some kind of nutritional quality. And I must say, there is so much more trials that were conducted that I can't bring here today for a question of time. They tried to tomatoes, lettuce, they have more success than the photo that I showed you guys before. But this is just how far I went. So take home ideas. It's really important to think about these things, like to go out, to have a backup humanity plan, but it's really hard. There's a lot of challenges on the way that can mainly hit the DNA and cause DNA damage. But there is hope. There is some kind of plants that can resistant against these situations. So it's up to research to find out why. And yep, thank you very much for your time. I hope you guys learned just a little bit, not too much. And yeah, thank you very much. If you want to find me, does anyone have any question?
I have a few questions for you. Can you go to page 22? Can you see the difference between Mars and ours?
Mars and what?
Ours.
Earth.
So can you see the difference how ours is green, how we have plenty of water? And then you see how Mars is dry and it's diesel. So do you think we can survive in Mars with less resource while we are ignoring Earth with so much resource?
Well,
just from this picture, you can see that like we have plenty of things on Earth, but we are ignoring Earth and we are focusing like on Mars. On Mars, we have many challenges. As you said, the distance is very long. And then we have less resource. We have too many challenges. One thing you didn't mention on your presentation, you know, Mars is very cold. You know that? And we even almost we don't have any water. We have very thin atmosphere layer. We have all these challenges. By the way, this is very great presentation. I like your presentation. It's very, especially as a master student, this is very top-notch presentation.
Thank you.
But even the research, the exploration, that's good. We can, when we explore for Mars, we can solve some of the challenge on Earth. But for me, I mostly ask myself why we are concentrating on Mars rather than protecting our planet, Earth. Yeah, that's my basic question. And my other question, when do you think we could be able to colonize Mars?
Okay.
Yeah, these are
What's your name?
Question.
What's your name? How?
Fitson.
I'm not very good at spelling. Just can you say it again? Fitson.
Yeah.
That's hard. Okay. Well, thank you for the questions. Are really good ones. And answering the first one, like why? Well, like I said in the beginning, there's a lot of reasons why, but actually, I think, in my perspective, that we are capable of both, like protecting our earth, doing agriculture and everything in a environmental thinking way, but we need a backup plan. We need to go farther away and try to go to other planets. It would be great if we have another planet like Earth, but the forecasts are for 2050 to get like 10 billion people on planet Earth, and you're going to need to increase the production of food for like 70%. So it's hard. It's possible, it is, but who can guarantee that we're going to stop at 10 billion? So we are going to need more space. It's hard, but it's just one possible way to give out of this situation. We can do both, in my perspective. It's possible, nobody knows, but it's my opinion. And when you ask it, I mean, I'm not an expert on this kind of topic, but NSA tells about, Elon Musk maybe tells about 2026 to go to Mars. I don't really believe it, that they're going to do it so soon, most like 2030, but it's just a guess.
Do you want me to guess when we can colonize Mars?
Sure.
We cannot colonize Mars in 1,000 years. That's my guess.
Okay.
Yeah.
Fair enough. Eu tenho some.
I have a question. First of all, nice presentation. I really enjoyed it.
Thank you.
Right now we're doing soils and landscapes with the shoes. And are there any extreme landscapes here on Earth that we can use to somewhat try all these things?
A very good one. Thank you, Nelson. And yeah, there is like, I read some of the articles that I read was actually trials on the desert of Sahara, which are really, really arid environments. Even with like the hot on the day and the cold in the night, they try to simulate a microgravity there. So to try to understand what is going to be like to be there on Mars, or just like traveling to Mars, it's already a really hard condition. The thing is that I read was like, you need to have like a perfect system. You can have ways on the treatments that they have, which would be like a city, a small city with everything that people need to survive. They had like 0.02 waste of the system, like when talking about carbon, oxygen, everything. Even though it's not enough to get there. So it must be almost perfect. So they try in this kind of arid situations.
Thank you.
Thank you.
Okay. Thank you, Bruno, for this great presentation, and thanks everyone for coming. I really love this presentation just because at some point I was really interested on this topic too, and I think it's just interesting how we as an agronomist can be involved in something like this, because at some point I thought it was not possible. But there's so many things that we can do as an agronomist from science. So I think it's really interesting. And also, I think when you were mentioning your question, I was thinking about when people was trying to get to the moon, and they are trying to do all this engineer to try to go to the moon. And all of those efforts help us in Earth to develop more technologies here on Earth. So I think we learn a lot from that. And if we have those type of challenges, I think we can evolve really well in science. So I think this is really inspiring. So thank you very much for this. And thank you everyone for coming. We will have another seminar next Friday. It's not gonna be here on this room, it's gonna be on the basement, but we're gonna send the flyer so you can attend. So the presenter will be Ana Morales.
Nice.
So thank you very much for coming.
Thank you. Nice set.
I have